At this point, you've learned about the basic structure of the cells that comprise the human body. We began by looking at the nucleus, focusing on its structure and function, then moved out to the cytoplasm and learned about each of the cytoplasmic organelles. To conclude our discussion of the cell, we'll focus on the plasma membrane, the outer boundary of the cell. Remember that all cells are surrounded by a protective plasma membrane, and this plasma membrane is incredibly thin. Despite its thinness, however, the plasma membrane carries out three very important functions. First, as the outer boundary of the cell, the plasma membrane separates everything that's inside the cell from everything that's outside the cell. So not only does the plasma membrane ensure that the cell's components stay within it, it also protects those components from harmful substances that may be in the environment surrounding the cell. The plasma membrane is also selectively permeable, which means that it regulates the exchange of substances between the cell's internal environment and its external environment. In other words, the plasma membrane chooses what can and cannot enter and leave the cell. So, for example, water and other vital nutrients can pass into the cell because the plasma membrane chooses to allow those substances to cross through it. Finally, the plasma membrane is important because it allows cells to communicate with each other. This can happen in a number of ways. For example, certain cells can fuse together as their plasma membrane, providing a direct connection between them. Once again, recall the figure of the composite cell and take note of the plasma membrane. As we move on and take a closer look at the plasma membrane, you'll see that each of the major functions just described is reflective of the membrane's structure. So let's look more closely at the structure of the plasma membrane. The plasma membrane is composed largely of phospholipids, which are organic molecules similar in many ways to fats. The figure on the screen shows the structure of a typical phospholipid. Notice that a phospholipid has two distinct parts: a polar hydrophilic head region and a nonpolar hydrophobic tail region. The structure of the phospholipid is important because it determines the overall structure of the plasma membrane. For example, in this figure you see a row of phospholipids with their hydrophilic heads and hydrophobic tails labeled. As the name implies, the hydrophilic region of a phospholipid is water-loving. In other words, because it's polar, that is, it has an electrical charge, it will interact with other polar molecules like water, which you can see surrounding the hydrophilic portion of the phospholipids in the figure. Notice, however, that the hydrophobic tails of the phospholipids are oriented away from the water. This is because the hydrophobic portion of a phospholipid is composed of long, fat-like chains, and as you know, fats like oil and water don't mix. So the tail region of this molecule does not interact with water. However, all of the cells in the human body contain water and are surrounded by it. So if the plasma membrane was composed of a single layer of phospholipids, as indicated in the figure, this would mean that the hydrophobic tails of the molecule would be forced to interact with water. As you know, however, fats and water don't mix. So if this were the case, the plasma membrane would be unstable and would consequently break apart. To avoid this, the plasma membrane is composed of two layers of phospholipids, oriented so that the hydrophilic heads of the molecules interact with water both inside and outside the cell, and the hydrophobic tails of each phospholipid interact with the hydrophobic tails of other phospholipids on the interior of the plasma membrane. Because the plasma membrane is composed of two layers of phospholipids, it's referred to as a phospholipid bilayer. The structure of the plasma membrane is actually much more complex than the simple phospholipid bilayer depicted here. This figure shows a much more accurate picture of what the plasma membrane looks like. First, notice that two distinct sides of the plasma membrane are labeled. You can see the extracellular environment surrounding the outside of the cell and the cytoplasm on the interior of the cell. In looking at the figure, notice that scattered throughout the phospholipid bilayer there are a number of proteins. These membrane proteins actually float throughout the bilayer, and because they move, the structure of the plasma membrane is often referred to as the fluid mosaic model. The proteins found in the plasma membrane can be divided into two groups. First, there are integral proteins, which span the entire width of the plasma membrane. So in other words, they extend from the extracellular side of the membrane to the inside of the cell. These proteins, like the one indicated by the red arrow, typically serve to transport substances into and out of the cell. There are also peripheral proteins, which are only found on one side of the plasma membrane. The protein indicated by the blue arrow, for example, is found on the extracellular side of the membrane. Also notice on the extracellular side there are glycoproteins. Remember that glycoproteins are first produced by ribosomes attached to the rough endoplasmic reticulum, and after passing through the rough ER, they're passed on to the Golgi apparatus where they're modified before being shipped to another part of the cell. In looking at this figure, you can see where that glycoprotein ended up. Also notice in the figure a structure labeled carbohydrate of glycocalyx. The term glycocalyx refers to a sugar coating found on the exterior surface of a cell. All of the cells in the human body have a very unique glycocalyx, and the glycocalyx plays a critical role in a number of processes, including cell-cell communication. For example, the unique glycocalyx of sperm and egg allows the sperm to recognize the egg and thus fertilize it. Research has shown that glycocalyx may also play a critical role in cancer. For example, cancer cells are able to evade the body's immune system by rapidly changing their glycocalyx. Once again, as you can see, the structure of the plasma membrane is rather complex. However, understanding the structure of the membrane is imperative to understanding the processes that occur within and across it. Before moving on, re-examine the structure of the plasma membrane as shown both on the screen and in your notes. Remember that one of the main properties of the plasma membrane is that it is selectively permeable, which means that it only allows certain things to move into and out of the cell. For example, water can easily pass through the plasma membrane, as can small nonpolar gases like oxygen and carbon dioxide. Large molecules like glucose and proteins and charged particles like sodium and potassium ions, however, cannot freely pass through the plasma membrane. Take a look at the figure on the screen. The blue rectangle represents the inner space of the plasma membrane, and the orange lines on either side mark the boundaries of the internal and external surfaces of the membrane. Notice that in the extracellular space surrounding the cell, there are two different molecules. The molecule on the left is much smaller than the molecule on the right. Let's assume that the molecule on the left is a molecule of oxygen gas. Because oxygen is small and uncharged, it can easily pass through the plasma membrane. Now let's assume that the larger molecule on the right represents a molecule of glucose, a sugar the body needs in order to produce energy. Watch what happens when the glucose tries to cross through the membrane. Because it's so large, when it reaches the plasma membrane, the glucose stops. It can't fit through the plasma membrane and thus can't freely enter the cell. Although glucose can't enter the cell by passing directly through the plasma membrane, this doesn't mean that it can't enter the cell. Molecules like glucose that are either too large to pass through the membrane or that have an electrical charge can cross the membrane and thus enter the cell with the help of a transport protein embedded in the membrane. So as you can see, the structure of the plasma membrane allows it to be selectively permeable. The transport of substances into and out of the cell may be either active or passive. In passive transport, molecules will move across the plasma membrane from an area of high concentration to an area of low concentration without the use of cellular energy. For example, look at the figure on the screen. Notice that in this figure, the molecules are simply moving from one side of the cell to the other. In active transport, molecules are pushed across the plasma membrane, and this push is provided by cellular energy in the form of ATP. Take a look at the figure on the right. The molecules in this figure can't cross the plasma membrane on their own. So before they can move across the membrane, energy must be provided in the form of ATP. Once the energy is released, those molecules can then move across the membrane. Remember that in passive transport, molecules move from an area of high concentration to an area of low concentration. This process is called diffusion, and as a result, molecules and ions become evenly dispersed in their environment. Again, the main factor driving this process is a concentration gradient. For example, when the concentration of oxygen inside a cell is lower than the concentration of oxygen outside the cell, oxygen will move across the plasma membrane until there is approximately equal concentrations both inside and outside the cell. In other words, oxygen will diffuse across the membrane until an equilibrium state is reached. To better illustrate this process, look at the figure. In figure A, there is a beaker filled with water, and to that beaker a purple dye tablet is added. In this example, the dye tablet represents the area of high concentration of purple dye, and the water in the beaker is the area of low concentration. As the dye tablet dissolves, the purple dye will spread from the tablet, the area of high concentration, through the water, the area of low concentration, until it fills the entire container, as is depicted in figure B. So to recap, diffusion is a passive process in which molecules or ions move from an area of high concentration to an area of low concentration until an equilibrium state is reached. There are several factors that affect the rate of diffusion, one of which is concentration. The important thing to remember is that the greater the difference in concentration between two environments, the faster the net rate of diffusion. To better explain this concept, look at the figure on the screen. Notice that in the figure on the left, there are more molecules above the plasma membrane than there are below it, whereas in the figure on the right, the particles are more evenly distributed across the plasma membrane. Because there's a greater difference in concentration on one side of the cell than on the other, in the figure on the left, those particles will diffuse across the plasma membrane relatively quickly to reach equilibrium. In the figure on the right, however, the particles will still diffuse across the membrane, but they will do so much more slowly than the particles on the left. So again, remember, the greater the difference in concentration between two environments, the greater the net rate of diffusion. Two additional factors that affect the rate of diffusion are molecule size and the temperature of the system. And both of these factors affect the driving force of diffusion, kinetic energy. Kinetic energy is the energy of motion or movement. So, for example, small molecules have much more kinetic energy than do larger molecules. In other words, it takes longer for large molecules to move from one point to another than it does for smaller molecules to move the same distance. So again, the smaller the molecule, the faster it moves, the faster the rate of diffusion. Temperature is also very important. The greater the temperature, the faster the particles move, and thus the faster the rate of diffusion. To illustrate the effect of temperature, look at the figure on the screen. The blue rectangle represents a beaker of salt water, and the salt particles are represented by the purple circles. The beaker is also at room temperature. And at room temperature, you can see how quickly the particles move. Now let's take a look at what happens if we take the beaker of water and heat it on a hot plate. As you can see, those particles move much more quickly than they did at room temperature. So by increasing the temperature, the kinetic energy of the particles increases, meaning that they move much more quickly, and as a result, diffusion occurs more quickly at this higher temperature than it does at room temperature. To recap, at this point we've discussed two types of diffusion. Simple diffusion is the process whereby the kinetic energy of particles drives them to move from an area of high concentration to an area of low concentration without the use of cellular energy. In facilitated diffusion, particles move from an area of high concentration to low concentration with the help of transport proteins that are embedded in the plasma membrane. Facilitated diffusion, like simple diffusion, is a passive process and does not require the use of energy. One additional term that you should know is osmosis. Osmosis is the diffusion of water through a selectively permeable membrane. In other words, osmosis is the movement of water from an area of high concentration to an area of low concentration across a cell membrane. Osmosis is important because it allows the body to retain water, keeping us from getting dehydrated. That concludes Part 3 of Lab 2, the cell. Follow the directions outlined in your lab handout for completing this week's lab experiment.